Hi, I'm Dan Stanton, the editor and founder of BioProcess Insider, and I'm delighted to be joined by Felix Xu, the Senior Vice President and Global Head of WuXi Advanced Therapies. Felix, welcome.
Thank you. Nice to see you again, Dan.
Always a pleasure. Now, we're going to look at the cell and gene therapy space, and many people have seen this space as really emerging only in the last few years and very, very new. I mean, WuXi, however, has been playing in this field for the past 20 years or so. So I wonder, Felix, if we could start by you telling us how what changes in innovation and services WuXi has seen since you first entered the space all that time ago, and whether that's kind of accelerated or changed with the success of the first sort of handful of advanced therapies.
Sure. So we started doing cell therapies back in 2004, and back then it was a very small business. There were a number of companies doing it, but it wasn't like what you see today. I think what we began to recognize is we began to see an uptick in the number of companies entering the money that was coming in, and even the different types of products. So I remember back in 2012 and also in 2013, we brought this up with the board and the CEO that this might be a space that we want to do some more investment in. I think they carefully considered it. We just kind of watched the market. And I think by the end of 2013, it just looked like this was definitely a market that we needed to be in and we needed to be a bigger player in. Because you started seeing things like CAR-Ts, you started to see more investments in the cell therapies, and you started hearing more and more about gene therapies. And so we started probably in 2014, really accelerating the investment that we made in the space. And that investment in the space was not just facilities. Everybody thinks about manufacturing facilities, but really building out the people in the space. So we went from, for instance, 3 or 4 people in analytical development to almost 40 people. We went from initially having a very, very small group of process development to 60 people today. So the investments not just come with facilities, the investments also come with the skill set and the talent that you need in there. And then the other part that we spent a lot of time looking at was just what's available in the way of innovation and equipment, raw materials, and things like that. I think the space really has exploded. And I think what you're starting to see in this emerging market is the market is struggling, I think, at times to keep up with the demand. And so you see that because there's capacity crunches. And that capacity crunch isn't simply in, everybody thinks about manufacturing suite, but it's also in process development. There's not enough process development talent. You also see it in analytical development, and you see it in raw materials. So certain raw materials at times can stretch out. There was a point where there was a key raw material that stretched out for 75 months if you were a new customer, okay? So I think all of those things kind of come with a market that's just really starting to take off and exploding. But along with that, I think what you're starting to see is you're not starting to see, you are seeing a lot of advances in the technology. And that's because the demands for the output have really picked up, especially in the gene therapy side. So I think what's happening here is there's a number of trends happening. One is that a lot of the early customers are getting to later phase. That means they have a much higher demand on production. You're also seeing, especially with things like AAV, if it's a systemic indication, you actually may need 200 liters for a single patient. Well, that's not going to be sustainable long term in terms of how, so the industry as a whole really needs to look at how do we increase our efficiency, how do we increase our yields. And we need to do the same thing monoclonals did. So if you look at monoclonals 30 years ago and their yields, and you look at the yields today, they're 10-fold higher. And I think this industry will go through the same evolution. I think the other aspect that this industry needs, and I think a number of people are looking to do this, including ourselves, is you have to have platforms in the industry. You can't have improvements if there's nothing to standardize and improve. So I think people are looking to build platforms. And once you have a platform in place, it gives you something that you can keep building upon. So once you have, okay, this is my process. So for instance, Bush right now has platforms in 4 areas. We have it around AAV suspension, lentiviral production, CAR T cells, a closed process, and then also around the manufacturing of TILs. Okay, those are areas that we really have built a platform around. Now that we have a process for manufacturing AAV, we can every year look at how to improve that process. So you can say, okay, this year we're going to focus on improving the upstream. Well, what can we do? Do we get better plasmids? Do we relook at media? Do we look at some other ingredients that we add? You may then at the same time or a little later say, we're really going to focus on the downstream. So how do we improve the yields that we get? Because yields in vectors right now, if you look at the literature, is anywhere from 10 to 40%. Well, if you can improve the yield to 60%, you just increased it 50%. So are there better resins that we can use? Are there other ways to do the downstream purification that improves the titers? And I think the industry as a whole is just looking at different areas like this to figure out how to do that. And then technology companies are coming in offering potential solutions to do these things better. But I think part of the critical aspect of improvement, though, is you do need to have platforms in place that people can improve upon. Kind of a, here's the process to do this. Okay, now that I have the process, how do I improve that process? In monoclonals, they eventually settled around the CHO platform. Now that they have the CHO platform, they've spent years improving how to do the CHO platform. And so I think in the same token, we'll look at that. We won't be able to get one platform for cell and gene therapy, but we may get one platform for AAV. We may get one platform for Lenti. We may get one platform for CAR T. So there's big areas that we might be able to get a platform that we can keep improving upon. So I think it's a pretty fascinating industry right now. It's pretty exciting, but I'll just put it this way: you have to be comfortable in an environment of uncertainty because things change very quickly.
They do indeed, Felix. So what you're saying there are that at the moment there are a lot of different platforms in play, but as time goes on, we're going to see them sort of whittle down to the successful ones, the most efficient ones. And at some point, hopefully in the near future, but you'll be able to tell me more about that, we will have a set of proven platforms for various different cell and gene therapies that will be available to industry. Is that the case?
I think that'll evolve. I don't know if it'll be in the next couple of years or if it'll be in 5 or 10 years. I think we're still exploring. I'll give you an example. You can look at AAV suspension. The demand for yields and output of there is tremendous, especially when you're systemic indications like Duchenne's or hemophilia. So right now, the platform that most people are using is HEK. HEK is good. It's got its own limitations on yield, and also it's a fairly expensive process. People are saying, is there a different way to do that? And so people are going to explore something like a baculovirus system or a producer line system. And if they can get that to work better, and it's something that can be more standardized that the industry can adopt, the industry may ship that way. Or what may happen is people find a way to do HEK much more effectively, more cost effectively, and that continues to be the system that people use more. So I think things will evolve out, and it kind of depends on what happens with these different systems. We're agnostic to that, and the fact that we're going to explore a couple of different options. We're not absolutely committed to one system versus all the others. Right now, we have our systems in HEK system, but we are actively exploring and looking at other systems also in the event that these systems are actually able to produce the product that's as effective at a much better yield and much more cost effectively.
What is interesting to me, and once again, correct me if I am wrong here, but on the innovation side, on the development of these platforms, it's the onus for small molecules or even biologics has previously been on the end user, but more and more when I'm reading about or hearing about cell and gene therapies in development, the innovation is being driven by third parties like yourself. First of all, is that the case? And if so, why is that?
I think it's both. I mean, if you look, I'll take baculovirus, for instance. The biggest drivers of baculovirus have been the customer and developing the system. I think, though, if you start to look at, let's take the AV suspension with HEK, I think CMOs have driven a lot of innovation there. And if you go back to the constraints in the industry, there's a limitation on process development, talent, et cetera. For a lot of small companies, they're much better off going to a CMO that's developed the platform than to try to learn the platform themselves to develop it. They may not be able to get enough people, plus they have very tight timelines. And so there's a real learning curve to this. So as an example, when we develop a platform, it takes us 12 to 18 months to develop it. So it's not something that we just sit in and one day decide, okay, this all looks good, let's put it together. We actually put it together, then we have to test it. Oh, you know what, that didn't work nearly as well as we thought, let's try this. And you end up doing a lot of that, and then part of that is you also want to say, okay, if this is optimal resin as an example, and it doesn't work for a particular customer, what's the backup resin? And so you're really kind of characterizing your process to a much greater degree than typically what you get from a customer, because a lot of times the customers, for instance, are inheriting it from an academic center where they, and so the academic center is put it together, they figure out how to make it, but they don't really have a characterized process. So I think, from that standpoint, we're not the only ones to do it. I'm sure there are other CMOs that have done the same thing because they've had the chance to develop them themselves and also to work with multiple clients to field test it for real. Their learning curve plus the background and baseline they develop is going to be much faster for the client than if the client tries to do it themselves. And then as you're doing that, you're just constantly trying to improve it. Because as a CMO, I'm competitive with the other CMOs. So I want my platform to be better than theirs. They want theirs to be better than ours. So we're all trying to see how we can develop ours as quickly as possible and as efficiently as possible. So I think that part of it, you will see CMOs taking a real strong role in, but I think there will definitely be customers that will have a lot of input on the innovation also.
And talking of those customers and talking about speed as well, speed to market, it's obviously a huge competitive advantage and a lucrative market opportunity. But how important is it from your client's perspective for this new class of therapeutics compared to, say, a monoclonal antibody or a small molecule in order to get their product to market as fast as possible?
I would say right now, speed to market is probably their number one criteria. With cost being the second criteria. My guess is 2, 3, 4 years from now, cost may come up to be an equally important criteria. But right now, I think with a lot of this innovation, you want to be the first one out to the market. You want to be the one that gets approved first, that puts your name and stamp on it, that people refer to as that's the treatment right now in this space. And so I think that's where people place a lot of effort and time. And if you look at, for instance, a lot of startups that get funded, they have very clear guidelines for where they would like them to be, start the clinical trial, first GMP, and those are pretty aggressive milestones, pretty aggressive timelines that they've been given. So I think all of that together does drive time to market as being probably the number one criterion will be for a period of time. And so that's where I think it's very important to, find the most effective way to move forward with your product. I think one of the difficult parts, if you're doing the process yourself, is there's just this learning curve around it. I think the other part is knowing when to stop tinkering with the process. And I think that can be difficult, and a lot of scientists will say that because they'll take it to a certain point and then they'll think, oh, if I did this and if I did that, process would be in. But at some point you have to stop to go into GMP. You have to have a fixed process. And again, that's why it takes us 12 to 18 months, because we ourselves are doing the tinkering for that period of time until we just kind of say, that's enough. The next round of changes, the improvements are important enough to justify the delays in the timeline. So I do think that whole part of it is important, because once you've got this and you can do it, I think the whole point of a platform for us, and I think we're not the only ones, is not to get the absolute optimal titer. It's to get a good enough titer in a fast enough timeline. So you're not looking to say, I want to shoot for this as high as I can titer. You want to say, what's an acceptable titer? And how quickly do you have a process to get me there as quickly as possible? And I think that's, for many companies more important than trying to optimize the titer right now, is how quickly can I get into GMP. And so I think companies that have developed a process and have a way for screening, and if it doesn't go right this first time, have a backup plan for the next steps, they're going to be able to get people to the GMP faster.
I think that goes to reflect what you were saying about the huge demand, and also the push for speed here, and balancing when to go forward with this platform, and when you're going to get when you're going to get a good enough product to progress. Do you think then that looking forward, when costs come into play and when the first of this wave of products go through the clinic and come out the other side, we're going to readdress these platforms and look to incrementally increase efficiencies and productivity, like we're kind of doing in the biologic space?
Yeah, absolutely. In fact, I think if you're doing a platform right now, that's already on your mind. Your next step is, okay, how do I keep increasing the titer? Because as you keep increasing the titer, then what that means is the costs keep going down for the client. So if I can increase it, double the titer, I've just cut the cost in half, theoretically, right? That's why increasing it tenfold is so important for monoclonal antibodies. I think the same thing for us. So we want to be able to look at how we can do that, or we want to, in addition, look at, is there another process that allows us to do it for the same cost or lower that gives us the same output? So I think that's a constant game that we're looking at, is once you have that first platform in place, then you start to look at, okay, how do I keep improving each part of that platform? Where to place the most focus? It's a Pareto, right? What has the biggest impact? You focus on those areas first to try to drive that. But you're also saying, okay, the market is talking about another technology to do this. At what point do we start evaluating that other technology, comparing it to what we have right now? That one's a little bit harder because it's much easier to continue to invest and improve a current platform than to develop a completely new platform. But you have to look at doing that, because if you don't do it and the competition does, and it works, you're going to get left behind. So these are all things that you're taking a look at. And I think that, well, I know we are, and I'm fairly certain that a number of our competitors we're also looking, continuously looking at how do we improve the yields and the performance of the product. So.
Well, I think it's going to be an interesting few years as these platforms develop and efficiencies increase, and obviously more and more products move through the clinic towards commercialization. Felix, I want to ask one last question, just shifting gears slightly here. Sure. WuXi Advanced Therapies, you're based in Philadelphia in the Navy Yard. That sort of area has become a hotbed of cell and gene development over the past few years. I'm wondering why that is, and how involved WuXi yourself is in the local scene there.
Yeah, so I think part of what's driving that in that area is just the excellence of the academic centers there. So if you look at, for instance, UPenn is probably, if not the leading center, one of the leading centers for gene therapy in the world. And so, if you look at what Dr. Wilson's been able to develop on the AV side, and what Carl June's been able to develop on the CAR T, I mean, these guys are leaders, academic thought leaders in this space. And what they're doing and what investors are doing is they're taking a lot of what they develop and other parts of the academic institutions in that area, and they're creating companies behind them because the technologies are really promising, et cetera. So I think that in and of itself, you see a similar thing in Boston. Boston's obviously much more advanced than Philly, but Philly, I think, is starting to see a lot of similar trends to Boston. I think where we've been able to help out is that we have been able to work with a number of companies that are in that space there. We actually work with more companies from Boston than Philly, but there's more companies in Boston. And for a period of time, we've had some key strategic partnerships, which also help to build this thing out. So I think that area is the Philly area. Also, you may have heard they've labeled themselves, and it's a good label, Silicon Valley. So that's CELLICON. And I was part of the advisory board when this first came out. I think it was a good marketing by the area, but they've called themselves Silicon Valley, and part of that's just driven around the technologies that are in the area for cell and gene therapy. It's going to continue to grow. I think it's going to continue to be a promising area. You're seeing a lot of other companies set up there, whether it's a company like Amicus or a company like Iovance. You have Sparks that's already in the area. You have other companies that have spun out from, obviously, UPenn also in the area. So I don't see that not growing even more in the future.
And with a nice marketing tagline such as Silicon Valley, I imagine it won't be too long till the property prices start reflecting those in Boston and Cambridge as well. Felix, it's always a pleasure talking to you. Thanks so much for discussing cell and gene therapies with me today. And well, have a great virtual bio.
Yep, thank you. My pleasure. Thanks, Dan.